implant study.
Uh, Thank you so much, ma'am. Thank you, President, Secretary, Treasurer, respected elders, and dear colleagues. I do not have any financial interest or relationship to disclose in the following one, which the patent is in office. So it is a corneal antimicrobial implant study, a novel in situ sustained release drug delivery system for infective keratitis. As we know, avascularity of cornea and poor penetration of drug are the barriers for antimicrobials in the posterior corneal space. And any other means by means of subconjunctival antibiotics or intravenous antibiotics doesn't reach sufficient concentration in the posterior cornea. And this is why we also go for a loading dose concept. On the other hand, excess use of antimicrobials, antifungals, and antibiotics are proven to be surface toxic, and most often they can mask the disease process itself. We don't know whether the disease is healing or not because of surface damages because of antimicrobials. But we know when we give intrastromal injection of antimicrobials, they work exactly in the nidus of infection, so they are more effective. So we know. On the other hand, a single intrastromal injection is eliminated very fast. And this paper demonstrates nearly 22 minutes of half-life of intracameral drug, and they also believe sufficient level of antimicrobials are not achieved in the stroma. They are achieved, but they are not long-lasting after an injection. So with all this background in mind, in 2014, we thought of having a need for a different drug delivery system, which is intrastromal sustained release antimicrobial implants, and that is justified by our ethics committee also. So the purpose was to demonstrate the efficacy of implantable intracorneal sustained release antimicrobials in posterior corneal infections and abscesses. It's a prospective interventional non-comparative clinical trial. Cases with posterior corneal infection documented clinically, microbiologically, and by corneal OCT were enrolled. A deeper corneal pouch was made either using laser assistance or manually, into which these implants were positioned. And results were analyzed every day using AIOS and slit lamp photography. Implants were removed in 3 to 5 days and re-implanted whenever required. They were also used in the interface during therapeutic DAL cases. We have two generations. One was Wartman 3 wafers, the initial generation. We prepared a stock solution, then a working solution, and 20 microliters were impregnated on the wafers. We dried it, stored it in an incubator, and we used it. The second generation, the present one, what we do is we use copolymers of polylactic glycolic acid, and we prepare it by means of hot melt extrusion technique in collaboration with the IIT initially, and now Microlabs and Sinai Medical Limited from Israel, they are supplying us. And we use 30 microgram disc of amikacin, 100 microgram for amphotericin, and 5 microgram for moxifloxacin. We also did in vivo in vitro correlation studies to predict in vivo responses. Antibiogram studies using MIC for release kinetics and time curve against different pathogens were also studied, and microbiological laboratories in Trinelveli and Coimbatore helped us in this. As I said, we filed it, and this is a prototype case of a fungal infection with the posterior corneal infection and an abscess documented clinically and with OCT. In this particular case, we made a pouch using femtosecond lasers, opened the pouch and implanted amphotericin B implant. And it was interesting to note on day one, there was complete resolution of hypopion and resolving abscess by after 5 days of removal of the implant, and after 7 days, a clear zone, a healthy zone is seen on OCT, and a very well healed case. This is. Similarly, we tried in bacterial corneal abscess. An amikacin implant was used to treat in this particular case. We also used it in the interface during therapeutic DALC. Here in this case, moxifloxacin 5 milligram moxifloxacin implant was used in the interface, and which healed eventually. This was a case of a post-DALC, this is a post-DALC case with segmental infection with gram-negative bacillus, where amikacin implant played a vital role in healing. Another case of nocardia in which twice we needed to implant, and eventually it also healed. Etiologically and clinically, 8 of our indications were fungal in our case series, 7 were bacterial, 1 was nocardial, 13 were primary deep corneal infections, 2 were therapeutic DALC interfaces, and 1 was a segmental infection which I showed. All the implants we removed in 3 to 5 days, and 2 of the cases we re-implanted, and we did not use any antimicrobials topically. And in all cases, infections healed. As I said, we also studied antibiogram studies, MAC for release kinetics. We studied moxifloxacin over Staph aureus, amikacin over Pseudomonas, moxifloxacin over Pseudomonas, moxifloxacin over strep pneumoniae, and amphotericin on fusarium, which is more common in South India. And we found an interesting evidence that all the implants released maximal concentration in less than 8 hours, and there was a sustained release over a period of 96 to 120 hours. So to conclude, this newer in situ sustained drug delivery system showed an optimal therapeutic effect in deep corneal infections in terms of prolonged corneal contact time, enhanced ocular bioavailability, patient compliance, and reduced surface toxicities. And this can revolutionize sooner. And these are some of the areas. This was in Eucornea. It was awarded the best paper of the year last year. This was in SOE, European Society of Ophthalmology, in AO for Global Award, ACRS, in ESCRS, and of course in AIOS. And presently, it's tough, but still we are going ahead with this research project, and Microlabs and Sinai Medical are helping us. We have already started our preclinicals and rest of the other works, and our collaborators are they have in fact filed in for the global patent also, and I believe we thought in 2021 we may get a product, but due to all these unseen COVID circumstances, it may get delayed a bit also. Thank you so much for the gracious invitation and to present this, and thank you for patient listening. Thank you so much.